Hello everyone, and welcome back to another episode of Teach Me in 10, which is, of course, Technology Network's video series where we're exploring topics which can be seen as otherwise pretty complex, and we break them down for you in just 10 minutes. Wonderful bite-sized episodes. And in this exclusive Teach Me in 10, we are joined by Dr. Marcos Herrera-Vaquero, who is the lead scientist for the development of IO motor neurons at bit.bio, which is a synthetic biology company focused on generating human cells, advanced research, and drug discovery. bit.bio applies its unique OptiOx cell reprogramming technology to rapidly reprogram induced pluripotent stem cells, or iPSCs, into any desired human cell type at an industrial scale while maintaining exceptional purity and consistency. To learn more about bit.bio, you can click the links that we've left in the description for you. But today, Marcos and I will be talking about bit.bio's latest cell type, IO motor neurons, finding out how they can help to overcome challenges faced by scientists studying motor neuron disease and ultimately improve drug discovery outcomes. So, without further ado, I'm so excited to say that you're about to watch this incredible Teach Me in 10 in full. Hi Marcos, how are you doing?
Hi, Lucy. Thank you. I'm great. And thank you for having me here.
Perfect. So let's get into the Teach Me in 10 and start our chat about motor neurons. So firstly, where are they in our bodies and what exactly do they do?
Yeah, so motor neurons are essential nerve cells in our central nervous system. They carry signals from the brain and spinal cord to our muscles to enable movement. Found in the motor cortex, brainstem, and spinal cord, they are divided or broadly classified into motor, upper, and lower motor neurons. Upper motor neurons start in the cerebral cortex and connect with lower motor neurons located in the brainstem and spinal cord. Then lower motor neuron axons extend from the central nervous system to target skeletal muscles, glands, and organs, enabling motor actions. This communication between muscles and lower motor neurons takes place at a specialized chemical synapse called the Neuromuscular Junction, ensuring proper muscle response to brain signals.
So then, if something goes wrong with these motor neurons, what would happen?
So, motor neuron death or dysfunction can lead to motor neuron diseases, which encompass a range of neurodegenerative disorders primarily affecting motor neurons. As these neurons deteriorate, communication between the central nervous system and muscles is disrupted, leading to symptoms like muscle weakness, wasting, and ultimately paralysis.
So then, what do you think are some of the examples of motor neuron disease?
There's a wide range of motor neuron diseases, but the most well-known or studied are spinal muscular atrophy, SMA, and amyotrophic lateral sclerosis, ALS. SMA is a genetic disorder that affects children characterized by the loss of motor neurons in the spinal cord, so lower motor neurons, resulting in severe muscle weakness and atrophy. ALS, conversely, is an adult onset condition typically diagnosed in the sixth decade of life, and it causes the progressive degeneration of both upper and lower motor neurons, ultimately leading to fatal paralysis.
So I know from the research that the challenge for patients is in the limited treatment options available too. Is that right?
Yes, that's correct. Riluzole, which is used to treat ALS, has been approved by the FDA almost 30 years ago and only marginally extends patient survival. So despite decades of intensive research and drug development, clinicians still have very limited options to treat these diseases. So this highlights the need for more effective therapies and deeper understanding of these diseases.
Absolutely. And then from that, to have a better understanding of conditions like ALS and to start exploring new treatment avenues, what methods are researchers using?
Well, researchers use various tools to study these diseases. Animal models, primary motor neurons, immortalized cell lines, and motor neurons derived from induced pluripotent stem cells, iPSCs, through directed differentiation.
And And then I'm wondering if you know of any challenges that they come up against using those types of methods.
Several and many. But the key challenge, I would say, is physiological relevance. There are also challenges regarding the sourcing of iPSCs and primary cells. And usually, these methods are very time consuming with limited scalability and batch to batch variability. And this is where a highly defined, reliable, and scalable and physiological relevant models are needed to advance research and develop effective therapies. And this is the model that bit.bio's human iPSCs derived motor neurons can provide.
Perfect. Okay, so you mentioned these challenges with scalability and physiologically relevance of cells, but I'm wondering if you can share with our viewers how bit.bio's human iPSC-derived motor neurons can make a difference and can help us to learn more about motor neuron disease.
Absolutely. They are a significant advancement for research. These motor neurons start as human iPSCs, but are transformed into motor neurons using Optiox technology. Optiox stands for Optimized Inducible Overexpression and addresses the limitations of traditional methods for obtaining iPSC-derived cells, such as gene silencing. This technology, and this is important, precisely controls how cells change into specific types, ensuring a consistent and reliable production of motor neurons. This technology has now powered the development of motor neurons, which express key lower motor neuron and cholinergic markers within days, and have a spinal motor neuron identity. They are user-friendly too. We deliver them in frozen state, ready to go once thawed, and they maintain their motor neuron morphology without clamping. Within 2 weeks, they show electrophysiological activity in co-culture with astrocytes, and they present a similar pattern to that of primary motor neurons. They are highly pure and consistent, which makes them perfect for research, and especially in supporting drug discovery programs. In short, because of this Optiox technology, they are a reliable tool for studying diseases more effectively.
So, what are the specific benefits of motor neurons?
Well, firstly, Lucy, they don't clump together when grown in the lab, ensuring a clean and consistent culture which makes experiments more reliable and efficient. They allow researchers, this lack of clamping allows researchers to perform experiments such as patch clamp to measure the electrical activity of motor neurons and high content cell imaging. Second, talking about physiological relevance, they accurately represent human spinal motor neurons right from day 4, showing key markers of spinal motor neurons. And by day 14, they start firing up spontaneously when grown with astrocytes, making them useful for studying diseases and testing new drugs. And this is crucial that they do fire spontaneously in the presence of other cell types, of astrocytes in this case, demonstrating the high purity of our motor neurons. Because of this technology and how consistent it is, and how well we characterize our product, you can expect consistent results every time.
Ideal. And then, before we wrap up, can you talk about the wider impact these motor neurons could have on the field of neurology and potentially even other areas of medicine?
Of course. So having motor neurons is a huge leap forward, not just in studying motor neuron diseases like ALS and SMA, but it also opens doors across neurology. They give researchers a solid, physiologically relevant tool, speeding up discoveries and treatment developments for many neurological issues related with motor neurons. This could go beyond just diseases. It might help us understand neuronal development, mechanisms of disease onset and progression, and they could be used in neurotoxicity testing. So really, these cells are not just about one field, but also contribute to advancements in various areas of medicine, opening new avenues for understanding, preventing, and treating a wide array of diseases.
This is incredible. It sounds like a real game changer. And as we're almost out of time, I really want to ask the question I'm sure everyone is eager to know. But how can people get their hands on them? Are they available now?
Yeah, they are available now through an early access program. They are frozen and ready to be shipped. And as an additional note, we've been talking about motor neuron diseases and ALS, and we are actively working on ALS disease models that will be launching later in the year.
Perfect. That's so exciting. Well, thank you so much, Marcos, for your insights on bit.bio's motor neurons and Optiox technology. It's been incredibly exciting to hear how these cells shape the future of motor neuron disease research. I appreciate your time and expertise on the topic.
Thank you, Lucy.
Perfect. And thanks to everyone here for watching this incredible episode. And I hope I'll see you again for another Teach Me in 10 very soon.